We are going to discuss about another electron microscope that is called a scanning electron microscope or SEM. This is the experimental setup of a scanning electron microscope, and this is the enlarged view of those objects which is viewed under SEM. What is the purpose of a scanning electron microscope? Scanning electron microscope is generally used for taking the surface view of object. In the previous class, we have discussed about what is meant by TEM and what is application of TEM, where we have seen that the TEM or transmission electron microscope is generally used for taking the ultra-thin sections of objects. Ultra-thin sections in the sense that they are generally used for taking the image of intracellular components, then cell organelles, cell components, plasma membrane, etc. But here, unlike TEM, here we are generally taking only the surface view or the morphological view of an object, not the anatomical features. The principle is somewhat similar to that of all other electron microscope, that is a very fine beam of high energy electrons having a potential of 3 to 30 kilovolt will scan the specific surface area of a specimen. Okay, so what actually we are doing is that the primary electrons are generally those electrons which is generally formed or which is generally produced by means of the thermionic process by providing a high input of volt or voltage into the cathode or electron. Whenever we are applying this much of voltage into the electronic gun, it will emit electrons. These electrons will soon impinges, or it will moves, or it begins to attack on the object, the microscopic object that we want to analyze. So it will move back and forth through the specimen. So what happens when the electrons will passes through, or it will make when it hits upon this microscopic object, what happens is that the electrons will be either scattered or reflected. Okay, because we know that in most of the microscopic object it contains atoms, molecules, etc. So whenever the electrons are impinging on these atoms or molecules, it begins to either scatter or reflect. And during this, the secondary electrons are emitted either from the surface or from the near surface area of the specimen. So what actually happens is that whenever this electrons get impinges on the specimen, it may be either this secondary electrons, that is the primary electrons is the electron which is emitted from the electron gun, and it will passes through this lens system and finally impinges on the specimen. And when it impinges on the specimen, from the specimen the electrons that means when it is moving out from the specimen, diffracted out from the specimen, that type of electron is called a secondary electron. The secondary electrons will be emitted from the surface of the specimen, and these electrons will interacts with the atoms of the specimen and eliciting detectable signals. So these primary electrons will be reacting with the molecules or atoms in the specimen and produces a secondary electrons, and the secondary electrons will produce some kind of detectable signals, and these signals provided the information. These signals will be fed into it either at a computer or a TV scanner. So the signal will provide an information that it will be the surface topography or external morphology, chemical composition or electrical conductivity and other aspects of that sample. What about the magnification of the SEM is that it generally causes 1 lakh times magnification than the usual object that we generally viewed under a light microscope. Then what about the application of an SEM analysis? This is a SEM analysis of pancreatic cells. Pancreatic cells are nothing but these are cells which are concerned with producing either insulin, glucagon, etc. So this is a SEM analysis of a pancreatic cell. And the application, one among them is it is generally used for producing high resolution image to reveal the spatial variations in chemical composition, how the different part of a particular object is different in their chemical composition we will get by means of producing high resolution images by SEM. Then another one is called the detection of phases based upon the qualitative chemical analysis or on the analysis of crystalline structure. We will get the detection of phases. Phases in the sense that when we are observing the things either qualitatively as well as quantitatively, we can identify the different phases of that chemical analysis or crystalline structure of a compound, in which stage it is generally formed, or in which stage it is generally under consideration. Then apart from that, the third one is called the precise measurement of the very minute features of the object down to 50 nanometer in size. That means we are able to identify the minute features which is having of those samples which is having a size below 50 nanometer in size. So that's all about SEM. If you have any doubt, please do in the comment section. Thank you.